That introduction, and I'd like to thank the organizers for the opportunity to have Syros present some of the work that we've been doing. And I'm excited to tell you about why we think fetal hemoglobin induction is a promising approach to treat sickle cell disease. And so red blood cells carry all of the oxygen to the tissues and to the organs throughout the human body. And the oxygen carrying capacity of these red blood cells is mediated through hemoglobin, adult hemoglobin. And in patients that have sickle cell disease, it's this adult hemoglobin that's mutated, which leads to defective adult hemoglobin, which I'm showing here, leading to crescent-shaped cells and ultimately manifestation of the disease. Fortunately, this mutated adult hemoglobin can be functionally substituted by activating a normally silenced but functionally equivalent gene known as the fetal globin genes. And this leads to functional fetal hemoglobin and normal red cell morphology, which I'm showing on the right. And the reason that we know this is to be true is through a number of genetic and clinical studies where a small fraction of sickle cell disease patients also inherit a second mutation known as a hereditary persistence of fetal hemoglobin mutation. And in these patients, the expression of the fetal genes has been upregulated and turned on, while the expression of the sickled gene has been shut off, and the mutated adult hemoglobin is dialed down. And what's really important about those patients is that they're largely asymptomatic if those fetal hemoglobin levels rise to a sufficient threshold. And so Syros' development program is centered around generating an oral medicine which can essentially do the same thing that's found in natural human genetics, upregulate the normally silenced fetal hemoglobin gene while silencing the adult and defective sickle gene. Oh, and that's an animation to show that. Silencing of fetal hemoglobin has been well studied by a number of investigators. And it's well known that fetal hemoglobin is kept in the off position in normal adult cells through DNA binding factors known as transcription factors. And these transcription factors are able to recruit other proteins which are able to repress and silence this gene. And while Syros is interested in understanding and investigating these known fetal globin repressors as part of a larger program around developing an oral therapeutic, the focus of this study was to ask the question, can we identify novel HbF repressors? And the idea here is that we might be able to understand more about the mechanism of silencing of fetal hemoglobin, but it also might lead to novel avenues for therapeutic intervention. And that's the subject of the talk that our scientist at Syros is presenting tomorrow, Dr. Mudit, Chaand, where he'll talk about Syros' effort in identifying a novel fetal globin repressor known as nuclear factor 1X, or NFIX for short. And so to make this discovery, we examined precursor red blood cells, which are known as erythroblasts, at various stages of maturity. And it's well known that erythroblasts that are derived from fetal tissues, such as cord blood, express high levels of fetal hemoglobin, which is shown in some of our data on the left. In contrast, erythroblasts that are derived from adult tissues, such as bone marrow, express very high levels of adult hemoglobin. And so we compared fetal and adult state cells at various stages, at discrete stages of maturity, to see if we could identify transcription factors that might be regulating the repression of fetal hemoglobin in adult cells. So using these cells, we applied a previously known technique, or previously described technique, known as chromatin accessibility mapping. And what this technique allows us to do is to visualize regions of DNA that are open or accessible to proteins. And the reason these regions of open chromatin, as we call it, are important is because they're often occupied by transcription factor proteins, and transcription factors are able to turn other genes on and off. And so by comparing these peaks from this chromatin accessibility mapping in adult or fetal state cells, we identified an enrichment for this sequence, DNA sequence, that corresponds to the DNA sequence that's normally bound by these NF1 factor proteins. And then using additional data that Mudit will talk more about tomorrow, we were able to narrow it down to the family member known as NFIX that might be the transcription factor that's leading to this fetal globin repression. And so we decided to test the hypothesis that NFIX is a fetal globin repressor by knocking down the expression of NFIX in primary cells and cell lines which normally express very high levels of adult hemoglobin. And by knocking down NFIX, we saw a very robust induction of fetal hemoglobin, which is shown in the data here. We saw nearly 100% of cells in which NFIX was knocked down express fetal hemoglobin. This level of induction compared very favorably to probably the two most potent known repressors of fetal hemoglobin, CBTB7A and BCL11A. We also found that the total amount of fetal hemoglobin in these cells was nearly 40%. And the reason that this is important is because the target levels of fetal hemoglobin suggested by hereditary persistence of fetal hemoglobin mutations in sickle cell patients is about 30%, and this is the level that patients become asymptomatic. So while this is obviously a preclinical investigational study, to see levels of induction in cells, primary cells and cell lines of 40% is very encouraging, and it shows that it's a very potent fetal globin repressor. And so in conclusion, Syros has a kind of a broad discovery effort in sickle cell disease, and we're focused on disrupting the function of proteins which normally silence fetal hemoglobin. As part of that effort, we identified and validated NFIX as a novel transcription factor involved in fetal hemoglobin repression. We see this discovery as potentially a new avenue for therapeutic intervention in sickle cell disease, but there's still a lot of work to be done, and our future work here is really focused on investigating if NFIX directly binds at the fetal globin promoter, or the region that normally shuts on and turns off the gene, and whether NFIX interacts with any of these previously known fetal globin repressors.